We are delighted to have Dr. Ishanu Chattopadhyay today, as good as I could do. And then he's an assistant professor of medicine at the University of Kentucky, specializing in artificial intelligence, machine learning, and computational data science. He leads the Zero Knowledge Discovery Lab, which focuses on complex disease etiologies and rare event dynamics in human and natural systems. His pioneering research has been recognized by numerous prestigious grants and awards, including the Defense Advanced Research Project Agency, DARPA, Young Faculty Award for his work on cognitive dissonance and opinion dynamics. His contributions have been funded by the U.S. Department of Defense, NIH, and the Alzheimer's Association. In today's talk, titled Third Wave AI for Medicine, from test-free screening of complex diseases to digital twins of microbiomes and pandemics, he will explore groundbreaking approaches in AI. These include test-free screening methods using predictive comorbid patterns for early disease diagnosis and digital twin models that aid in understanding complex biological systems and pandemic prevention. Dr. Chattopadhyay's work has been widely published in leading journals, including Nature Medicine, PNAS, Science Advances, and JAMA, marking him as a prominent figure driving AI innovations in medicine. And we look forward to your exciting talk.
Okay,
welcome.
Great. Thank you so much. Let me first begin by thanking for inviting me here and the immense hospitality from Brittany and Lectures. Thank you for that. Okay, so let's get into it. So I have broad research interests in AI, so it's always challenging to kind of talk about all of that and fit it in like 45 minutes. So I'll try my best. So third wave AI, what does that mean? What's the third wave? So that phrase actually comes from DARPA and DoD, where they try to categorize the different phases of AI. First wave in 1980s to 1990s, where you had rule-based expert systems. And second wave AI is pretty much what we have come to love. It's like big data, machine learning, deep learning, but they are not very they don't generalize well from the tasks they're trained for. And third wave AI is what has been emerging recently, 2023, is when I say, I kind of think of when AI went mainstream. You have contextual reasoning, you have generalizable models, and you're really stepping towards true intelligence, right? So that's the recent one that we've seen, the recent kind of emergent technologies of third wave AI. So my group kind of sits at the intersection of mathematics, computer science, medicine, and social sciences. And we develop tools in that context and think about algorithms that apply in that context that can help solve problems in clinical analytics, in biological and biomedical systems, social systems. And I also think about what the broader role of AI and machine learning is in society, the implication of where this is going, and really how we can apply these technologies to better understand, control, and think about complex systems. So this is some of the people in the group who I've worked with. They're pretty much all of them moved on to do bigger and better things. Just pointing out that the postdocs, Nicholas, Shahab, and he were all math PhDs, and most of the rest of them who are grad students, some of them undergrads, were essentially majoring in computer science, math, or economics sometimes. So while I was in the Department of Medicine, everyone in the group were really computer scientists or mathematicians. As I said, I have broad interests, and this is some of the collaborations that we have had over the last 4 or 5 years. And you can see the only reason I'm putting it up there is to show you the breadth of what the tools that we have developed, how they get applied. So you have Department of Pediatrics, School of Molecular Engineering, Department of Neurology, and it goes beyond Chicago. It goes to sometimes in UK, for example, we had some collaboration with the Alan Turing Institute, or the Dalhousie University in Canada. Pulmonary care, Center of Health Statistics. So it's a pretty broad footprint. And here are our collaborators: Robert Gibbons in biostatistics, Dr. Mastriani in neurology, Matt Honninghake in pulmonary care in Harvard, James Evans in sociology. So really, and these are only like a subset of that, more prominent people in their fields. And we have had this, and most of these collaborations are actually ongoing right now. So that pretty broad footprint. Where did the money come from for doing all the work I'm going to talk about? They were mostly funded by DARPA. I've been the PI of 3 DARPA grants, and site PI, one of them, which was the preempt one from the Biological Technologies Office. The other 2 were from the Defense Sciences Office and Information Innovation Office. I think that's what 2 I stand for. That was a D3M. All of them are essentially all AI proposals. D3M was data-driven discovery of models. PAI was physics augmented AI. And preempt, I can't remember what the acronym was, but it was about preempting pandemics. It was kind of funny because it started in 2020 and we had to take the PI meeting remote because there was a pandemic which no one predicted, particularly us. And the YFA is a Young Faculty Award that we got from DARPA. It's also on DSO. We have an ongoing grant from Alzheimer's Association, and we have also been funded by NIA for on the same kind of topic of predicting Alzheimer's, screening for Alzheimer's. Here we have been published in the last about 5 years, Nature Human Behavior, Nature Communications, Medicine. I really like this part of it, which is an interesting way of showing impact, which is the altmetric donuts. And you can see that at least one of the papers went over 1,000, which is pretty interesting. We'll not be talking about that paper though, because that's for predicting social interactions and crime in urban scenarios, urban spaces, which is kind of off topic from medicine. But we have important work in that aspect as well. So to talk about the other point I probably forgot to mention, I call my lab the Z lab. The ZZD stands for Zero Knowledge Discovery. The zero knowledge is for is indicative of the fact that we like models where there are almost no hyperparameters or magic numbers. So that's what the zero knowledge stands for. And the research trust, I have broad interests, but at an abstract level, there are kind of 3 key ideas that get applied broadly. And one of them is point-of-care screening for complex diseases. The other is a general framework for inferring digital twins. We'll talk about those 2 mostly. I'll not be talking about modeling, predicting complex social interactions, because, well, we're out of time. Okay. So test-free screening, that was in the title. What does test-free screening mean? Hold on to that for a second. Let's talk about universal screening. So we lack universal screening for most diseases. For example, there's no real universal screening for autism. There is a screening like MCHAT, which is really bad, and there's no one has been screened given a diagnostic test for autism universally. There's no universal training for idiopathic pulmonary fibrosis or Alzheimer's disease. Again, the cognitive assessments one could do, but it's not done universally. Suicidality, PTSD, cardiac events, melanoma, rare cancers, most diseases do not have universal screening. And the reason is mainly because of non-existent biomarkers or expensive, time-consuming diagnostic tests. Lack of universal screening at the point of care means early diagnosis is difficult, late or misdiagnosis happens, and that costs lives. Can AI fix that? And we'll see, we'll talk about how we kind of address some of those issues. We'll talk mostly in the context of one disease, the interstitial lung disease or pulmonary fibrosis, mainly because, well, again, I'm trying to compress on time, and we'll see how some of the tools that we have developed helps in early diagnosis, universal screening, and also almost automatically it allows one to do better cohort selection for clinical trials. We'll see that how you can actually do a continuous, a semi-continuous risk monitoring and track patient journeys as they get their risk increases and it eventually gets diagnosed. And I'm not going to talk about the last one, which is perhaps the most important, interesting one to me at this point, because that is still under development. but kind of predict prognosis at point of diagnosis. For example, adverse effects of interventions when somebody gets diagnosed with pulmonary fibrosis. So, okay, now let's talk about this notion of test-free screening. What do you mean by that? We mean the following. When someone walks into the primary care, without they being suspicious that they have a particular condition, or the doctor has been suspicious of that, can they use the data that is already there in their patient file, analyze that using AI, and make a reliable referral that this guy needs to go see a pulmonologist, that guy needs to go see a neurologist, and so on. So remember, the constraint is that you cannot ask for new tests. That doesn't happen. That's not something I'm imposing just for giggles. That's an actual problem. When somebody walks in a primary care, and if you need a brain scan to know if you have Alzheimer's disease, at the point when somebody is actually getting a brain scan, people already suspect something is wrong. That will not address this issue of late diagnosis and misdiagnosis. So to do it with data that's already there. That's what it means, test-free screening. So something like this. You start with primary care, you analyze the data, and you refer to a pulmonologist. Flag patients before they or their doctor suspect anything. So it can be done generally, and not just in the context of specific diseases. We have different models, but you can quickly train models to target different disorders, as I will talk about in a moment. We call that zero burden comorbid risk score when it has this platform, or Z-core. The zero burden comes from the fact that it's test-free. Basically, apparently the burden of testing is, it's not really zero. There's still effort going in to do this, but it's near zero. Now let's talk about a specific example, which is IPF, or idiopathic pulmonary fibrosis. Now IPF is not a disorder that one would like to have. It is something, the post-diagnostic survival is about 4 years. It's not very common, thankfully, but still about 200,000 people in the US have the disease. Affects more, like older people, greater than 50 years, affects more men than women, and it has very non-specific symptoms, shortness of breath, cough, and which is necessarily doesn't jump out and say that this guy has IPF, which means that results in at least lots of misdiagnosis and late diagnosis, and well, that reduces makes patient outcomes worse. Question is, can we make it better? That's what our study did. We looked at, it was a retrospective study of about 3 million patients, and it was a collaboration between UChicago, where I was at that point, with Mayo Clinic, Harvard, and Weill. And what we were able to do is to pull back the diagnosis from point of current diagnosis to about 4 years before. So that gives you about like 6 to 8 years time, if you can actually deploy this, before things get really bad, which means you could actually, the patient would be at a much better odds of getting a lung transplant, which currently is only treatment. But in future, there are things on the horizon which would be disease-modifying drugs, even for those who would be super critical to diagnose patients early, and this kind of technology would be incredibly useful. So before we go farther, one point that I want to draw attention to is that what is the kind of emphasis in AI research? It's mostly genomics, imaging, and drug discovery. And even when people are trying to look at EHR data to make screening tools, they are looking at the same data that a physician would look at and try to predict the decision. At that point, you're modeling the physician. You're not modeling the disease. So what we do, and this is not the only ones, we are not the only ones who have this idea, there's some other competing work as well. We look at the time series of all these codes showing up, the diagnostic codes, the medications as they're prescribed, the procedures, and kind of look at patterns that are emerging there, do pattern discovery, and try to get to the comorbidity fingerprints of that particular disease, which is essentially trying to find something intrinsic about the disease itself, and not just modeling the physician. So how do we do that? We set up a classification problem where you have cases and controls who don't have the disease for a sufficient period of time, cases where their medical history shows up with a code of IPF or ILD if you're targeting the disease broadly, and then you solve that classification problem, right? Easier said than done. I will not go into too much details of it, but I will talk a few things about what are the challenges of doing that. But even the specification has a problem, because just because somebody has an IPF code doesn't mean they actually have IPF. It might be just marked like an error or something that was put in because it was not realized at that point it was a different ILD and not IPF. So how do we make sure that our specification is correct? Well, we can do something like this. We can look at target codes, and we can look at prescriptions of IPF drugs, antifibrotics. It's unlikely that if you have IPF code and antifibrotics that you don't have IPF. Still possible, because IPF drugs are sometimes used to treat rheumatoid arthritis, but kind of less. You get more and more precise. Or you can add in signature of a diagnostic odyssey that will precede a proper IPF diagnosis. The reason is we cannot take deeply phenotyped data and learn our model from that. This is why we do all this. Because we need millions of patients before we can learn a reliable AI model. If you give me 200 patients, I cannot do it. We do not have millions of patients who are deeply phenotyped to know these guys do have IPF. No such data set exists. If you're looking for claims data sets, like market scan data on which this is done, you will not have that level of certainty, and you have to do stuff like this to make sure that you are getting more and more certain. So where does the data come from? As I said, the proven market scan data, which has about 100 million patients. We were able to extract about 2 million for the study from there. It is the Chicago Medical Center, about 70,000 patients, and a random sample from Optum Labs from Mayo Clinic, who kind of about close to 1 million patients. So, so in all, we looked at about 3 million patients. And here the results, I think to this audience is kind of pretty self-explanatory. That's the ROC curve, trade-off between specificity and sensitivity. And why there are multiple lines? Because you look at all these different subcohorts, males, females, high-risk groups, low-risk groups, defining cases as with codes only and codes plus antifibrotics, all of that thing. So the goal is to see if all the performance, no matter how you slice and dice and define them differently, are they all similar? And the result is yes, they are very similar. So that's like the most, the strongest kind of, we are happy to see that's how it turned out to be. So you get AUCs close to about 90%, and you have a likelihood ratios about over 30. So which means that if the algorithm flags someone, then their odds of getting IPF diagnosed within a year is about 30 to 40 times more than the baseline, right? The other key point to take away from this is that look at the low-risk cohorts and the high-risk cohorts. They're pretty much same ROC curve. What are the low-risk cohorts? Low-risk cohorts are patients who have no known comorbidity of IPF. So that's the most important subgroup for this analysis, because those people will 100% be missed. There's no way they'll be diagnosed in time without this technology. And let's wrap our heads around that this is not just finding new risk factors. You are looking at personalized risk patterns. Here are 3 patients, A, B, and C, who were diagnosed with IPF and were correctly flagged by the algorithm. And you see what their risk factors, if you can call them that, was used by the algorithm and how that changes from patient to patient, or how the weights on that changes. For example, COPD is an important comorbidity for IPF, but only 2 of the 3 have COPD. Cough is an important comorbidity of IPF. Only 2 of 3 have that. All of them have abnormal breathing, just extremely non-specific. Just because someone has abnormal breathing, you cannot jump and say they have IPF. And if you can do this, pick up on patients who are at very early cases of stages of the disease, you can automatically cut down on screen failure rates of clinical trials when they try to select early stage patients. This is a huge problem. Currently, the screen failure rate in clinical trials in IPF is about 60 to 70%. Sometimes it's stated as 50, but it's really around 66%. If you use Z-core to select patients, you'll cut down the screen failure rate to about 20%, which would imply that a cohort sizes of over 2,000, which is roughly the cohort sizes generally used, you're going to save the CROs about $20 to $30 million. So that's like a huge cut down in cost of clinical trials, which means you can do more clinical trials, which accelerates our path to actual cure. And patient journeys, you can run, if you have been following what I've been saying, no new tests are done, which means you can analyze a patient's data without the patient in the room. You can do it passively. At any time they are going to any clinic and their medical history has been updated, you can rerun it and see what the risk is. If there is nothing coming up, completely silent, that also informs the process, right? So basically, you can actually see how their risk is changing over time. Here are two patients who are diagnosed with IPF, and you can see how their risk went from 2015 to about 60% to about in 2019 to about over 90%. And you can see how the risk fluctuated. So you can actually chart this patient journeys, and insurance companies can actually do this in their entire patient population repeatedly to kind of leverage these underutilized diagnostic modalities and have kind of proactive interventions. There's a lot of stuff that needs to happen logistically and ethically for this to actually work, but we have the technology to do it. So now, kind of, it should be clear that whatever we're talking about, it is not specific to IPF. You can have the same technology to target different diseases. In fact, what we started with was autism. And when we published the paper in autism, which was in 2021, autism was 1 in 59, now it is 1 in 36. And we have a very poor screening tool that is currently deployed, MChat, which has a really poor specificity, really poor sensitivity. And by using ACOR, we call it ACOR at that point, which is Z-core for autism, you can show that you can actually either half the number of false positives or double the sensitivity, which would be super cool, super useful to reduce the number of people in queue for the diagnostic, the gold standard diagnosis that is required in autism to trigger services, and that would dramatically improve outcomes. You can do the same thing for Alzheimer's disease, and I understand there's a lot of interest about that here. And so using EHR data for Alzheimer's disease is not a new concept, but the papers that have been published have around 67 to early low 70s AUC. By doing stuff that we are, like our approach, it kind of boosts us up to about 87%. And there's nothing new, more like different to say about it. We do the same thing. We look at the EHR data, we look at the timestamp diagnostic codes, and we use the Z-core platform to find a model that tells you how whether someone in near future will get diagnosed with ADRD. And here's some classical results that exist. These three plots are about that. As you pull out when your diagnosis you are trying to do, whether it's like 1 year before the clinical diagnosis, 2 year, 3 year, 4 year, 5 years, 10 years, and you can see the competing results are rapidly decay, where the Z-core platform remains interesting, even if you're predicting 10 years out, which means you're actually capturing very early stage disease. You also actually target MCI, or mild cognitive impairment. The performance is not as stable for that, but it's still useful, definitely for picking up of MCI right now, and it is over 80% when you're picking it up 3 years in the future. So that is like way more early stages that you're picking up than what you can do even with that MoCA. Because MoCA does about 90% now. If you pull out 3 years, all bets are off. It's not going to be that predictive. And it's an ongoing list of diseases we can target with this. We have a set of publications on this: the autism, National Medicine for IPF, JAHA for cardiac risk after hip knee surgery. The Alzheimer's one is under review, but there's an increasing list of diseases. You have bipolar disorder, you have chronic kidney disease, you have rare cancers. You can do the same thing with suicidality, and that's something that we continue to develop. So before going forward, a couple of points. So I said that we have a different take on what the underlying AI system is, and we can talk about what's the secret sauce is. But does it really work the same if I use an off-the-shelf AI? And we have a detailed comparison in the Nature Medicine paper, and we can see that, no, it doesn't really work that well if you take and use something that already exists. In fact, it works well if you are restricting yourself to one database, but if you go into the other database, the performance goes down. And this is also true for if you have held back data in the same database. So it is not just overtraining, it's something different, picking up some features which are specific to that one particular database, which doesn't generalize something else. So that's going to be a problem. So how are we doing this? So what we realize, and whenever I give this kind of talks, I while talking about all the results that, that we have produced. That's definitely the main part of the talk. But I also want to talk a little bit about the insights that we have picked up along the way, which continues to inform our work and hopefully can spark ideas in other groups as well. So here's one of those ideas. So why can't we use a standard machine learning tool like LLMs to do this? Why do we have to come up with anything different? Well, so the reason is, if you are using an LLM, which is really large capacity of the model, you don't have enough data to train it. And if you use something like a support vector machine, which is very simple, low capacity, then it cannot actually represent the patterns necessary to have the maximum performance. So basically what we did here is that we have the cases and controls. We looked at odds ratios of all different ICD codes and a bunch of other engineered features, and we have a bag of odds ratios which we combine with a machine learning model to get the estimate of the risk. Now the question is really, what is the machine learning model you use here? So it turns out you have to control the capacity of that model. And what it turns out works very well is a network of LGBMs. If you use LLMs, too complex, doesn't train well. If you use support vector machines or something like that, well, it is too simple, it doesn't represent enough patterns. But at some point in between, you have the right capacity of the model that maximizes the performance. There's also the ability, we have the ability in the model that we use is to track longitudinal stochastic patterns explicitly. The way we do it is something like this. You think about this timestamp diagnostic codes, we kind of take that and compress it into a specialized HMM, which we call the probabilistic finite automata, which is work that we did about 10 years back now. And the way this works is that you look at the control patients and you look at the cases, and we call it positive here, and you look at different categories of diseases. For example, look at infectious diseases. And you look at every week whether there's a report of an infectious disease or not. So that will give you a binary stream. And you make this HMM, specialized HMMs for the control group and the positive group. And you get a bag of models for the control group and the cases. And then the question is that if you have a new patient with a data history, was that history generated by the control bag of models or the positive bag of models, the case bag of models? So how do you do that? I mean, it's non-trivial to compare HMMs that look different, have different structures. And there we kind of use this body of work that was reported by us in 2023. We call that a sequence likelihood defect. It kind of gives you a metric like KLD, the KL divergence, but instead of comparing distributions, it compares stochastic processes. So what's the state of technology right now? Well, we took about 3 and 4 years to do the theoretical formulation, the multi-core validation. Many of them are retrospective. There's some pilot studies going on. And we actually spent last year to make this into a user-accessible platform, right? If you have data like this, you bring your data, patient data, which is like, it could be a FHIR protocol as well, but like think of it as a JSON file right now, where you have time-stamped diagnostic codes, procedural codes, medication codes, and you can just use our API, which is deployed currently, and ask, tell me the risk for IPF, tell me the risk for dementia, tell me the risk of suicidality, tell me the risk of CKD, and essentially give you a computer value. It takes about 20 seconds, 10 to 20 seconds per patient. So it's kind of still not as fast as it can be, mainly because, well, it's a Google Cloud implementation. But it's kind of still, it gives you a nice demonstration of things. So one quick demonstration I want to do. Let me see. It will not show up there, will it? No, it won't. Okay. Let's see.
Required to change which application you are sharing.
Yeah, I know.
Maybe the PowerPoint.
Here.
Sure. If you press, oh yeah, there it goes.
Okay, so this is a live web page. You can actually go to there and upload your own data. and the API key would generally be required, but it is kind of keyed in right now. And suppose I say ADRD, and I upload a file of ADRD patients, about 100, and I load that patient file. It will take a second, hopefully. And yeah, so those are the risk factors, and it's on the left. You see that since you said look for ADRD, those are the known risk factors for Alzheimer's disease, and it's telling you what are the distribution in that data set that you uploaded with about 101 patients. And you can actually go and select to give me like all the patients that you have where the probability of true positive is above 97%, and kind of returns you all these patients here. You can download that. It's kind of designed for a demonstration to for clinical research organizations to kind of identify patient cohorts with much lower risk of string failures. But you can also do something really interesting. You can ask, why was this particular patient is positive? Why do they have that kind of high risk? So it kind of shows kind of a visualization of the decision path. It kind of says that this thing, your tick marks, are the codes that are there in the patient history, and having a particular code can either increase the risk or decrease the risk. Having E87 decreases the risk. Having I20 increases the risk. Also, there could be codes that are absent, like E87.8 that reduces the risk, and F1023 that increases the risk. So instead of actually risk factors, it's really like a two-dimensional thing where it can be present or absent, and having presence or absence can increase or decrease the risk. So I'll go back to the thing, which I have to stop sharing again. This one on. And then come back here. Okay. So that's kind of, I think, is pretty interesting. You can actually go to the Z-Core demo and actually run your own thing with it if you want to. So that's the direction one with the test-based screening. I'll talk about this is direction two and general framework for inferring digital twins. Let's go about that. The basic idea for that, in case I run out of time at the end, is to have something like this. I call it the bio-NORAD. So currently, lots of different strains of, say, influenza are being collected all over the world. We are mostly paying for it, so we're spending billions of dollars on it, and nothing comes out of it, really. Well, some research is done, but they're generally stuck in the fridge, in the freezer, and you don't really know whether the strain you collected is going to be the next H5N1 pandemic. So what would be great if we can actually detect that, if we actually predict that this particular strain is very close to emergence. How do we even model that? So that's kind of the question that we had in the DARPA preempt program. We wanted to know that if there's a strain in an animal, and spillover always happens. Pig farmers and pig farms always get infected with swine flu, but they don't go pandemic. So what is it that will actually make things go pandemic? That's the question. And so it took us about 4 or 5 years to figure that out, but actually there is a good approach at least. I'm not saying to completely solve the problem, but there's a good approach to it. So just to come so that we're on the same page, this is what like a cartoon of an influenza virus particle looks like. You have the internal genomic things, and you have surface proteins, and one of the surface proteins looks like this, which is hemagglutinin, the blue ones, which are involved in cellular entry in the host. So basically, the top part of the hemagglutinin actually interfaces with the host cell, and that is also recognized by the host immune system, and that's why the top part changes, varies, there's antigenic drift, so that it can elude the immune system, right? If all of this remain the same, you won't need any vaccines. The immune system is good enough to take it out, but they change under selection pressure, which makes it harder for the immune system to find it, and that's why this ongoing infinite game that's going on. So how do we know? I try to model what those changes are. The changes are not random, because this part might switch and change, but the function has to be preserved, or the fitness has to be preserved, so for the virus to actually be functional. So, which means that if there's some changes somewhere, there's some different changes somewhere else, where there's this association between them. There's a crosstalk happening, and I want to model that crosstalk. So how do we do that? Well, think of it like this. You have different virus strains. You line them up. They're all collected in 2020 and 2021. And you look at index 223, this one, right? And you'll see they are varying. They're not the same amino acid. They're not the same amino acid letter there, and it's kind of changes. So you want to build a model to predict what happens at index 223, and this is the model for that. You see that it kind of builds a predictor. It doesn't have to be a decision tree, it could be something else, but let's, for the moment, assume there are all decision trees. It uses as features which are different indexes. So for example, it uses 155, 89, but then 155 will have its own predictor, which will have other features, which will have its own predictor, and it will be a cross-linked recursive forest of decision trees. We call that a QNet. If you're interested, you can ask me later why it's called a QNet, but not important. So this is what I'm talking about. This is not just one decision tree. This is a hyperlinked decision tree which goes on forever. For example, for 220, this is the predictor. Each of those nodes actually you can zoom in and we'll get you a different decision tree. And this will continue forever. And this is all learned from the 400,000 sequences that we have for influenza in public databases, right? It goes on forever. So that's what the structure is. And this is the structure of the crosstalk. And this is a snapshot of all the models. It's not the 5% of it. So one challenge is visualizing all of this, but this is what it kind of looks like. Now, what do we do with it? That's a more interesting question. Fine, we got this model of crosstalk. It's a recursive net, and it actually has a few million parameters to it, depending on how much data you have. So it's really a large, I like to call it a large science model, not a large language model. It's not learning languages, it's learning the science of the crosstalk. Okay, so what do we do with it? What we do with it is that you can use this to define a distance metric, theta xy, which is a similarity between two strains. If you're aware of edit distances, they're not very biologically meaningful. They're like how many edits it takes to go from one strain to another. That really doesn't mean anything. Whereas this one is biologically completely meaningful. Not only that, if you find a distance that changes with the strain, it also changes with the background population in which they're embedded, because the speeds are actually functions of the QNet that we learned. Which means that if you take two strains, their distance would vary depending on whether you're asking they were collected in 2020, or they collected in 1990, or they collected in 2024. The background selection pressure changes the distance. It's informing what the distance is. Also, the distance is special. It's not just, there are a million ways of defining distances, right? But no, this is actually special, because you can actually prove that this distance scales as a log, a likelihood of the x jumping to y. So if you perturb it, valid perturbations, which means it produces valid progenies, and the probability it produces a y from the x scales as theta xy. You can actually prove this, right, from basic results in information theory. Now, one point, again, second insight that comes from this is that what you just did by this is you are connecting the metric structure of the system with the dynamics, like technically the tangent bundle. So you're saying geometry, you found a way of uncovering the geometry of the system that drives the dynamics. The other place where this happens is the theory of relativity, where you talk about the geometry of space-time, which drives how things move, right? It's kind of very similar. You're finding a geometry of the system, and it tells you how things are moving in the abstract strain space. I can go on and on that lines, how you can derive field equations from that, but I will not do that. The thing that kind of, so a couple of things. How do you validate this? Does any of this make sense? So the first kind of qualitative validation is that where are most of the differences coming from? If I perturb a sequence, strain y, and if I look at how the distance between the perturbed strain and the actual strain changes, where are the indices where the most of the changes are coming from? That should be the RBD, the receptor binding site. That's exactly what it is. It's kind of mostly concentrated on the receptor binding site. But there are other places where the changes are actually driving distance between them. So it's kind of matches with the intuition that virologists had, but it's just more precise. Okay, can we do some quantitative validation? Well, this is one way of doing it, which is CDC has about 24 scores of emergence risk. They look at 24 strains in 14 years. And the way they do it is they bring together subject matter experts, they do animal modeling and all of that. Takes very long and very expensive. That's why 24 such strains were analyzed in 14 years. And we looked at what happens if we compute our risk in the following manner. We have a distance, we have particular animal strain, and we were circulating human strains. What if we say the risk is when the distance between this animal strain and the average human strain is small, which means there's a higher probability of jumping. If we define that risk, we are not using CDC's data. It's completely independent evaluation, and this is what it looks like with R of 0.72 and p-value of 0.0001. So it matches up really well. Two completely different approaches of looking at the same thing, and they match up perfectly, almost perfectly, as perfect as you like, I think. And now, also, this evaluation takes 1 second instead of that taking like 6 months per strain. And if you can do it, then you can, in future, realize something like the BioNORAD, where anyone collects any strain anywhere, uploads a sequence in the server, and immediately lights up at what the risk is. And maybe we can go and inoculate that animal at that, for that strain and stamp out the next pandemic before the first human infection. Okay, I'm going to skip this because I'm running out of time. You can do the same thing for modeling ecosystems in measuring human microbiomes. That's a paper that came out in April. Looks at this kind of data, which just looks like a jumble of interactions and makes a QNet out of that and makes really nice predictions of how the different bacterial species fluctuates over time. So there are a host of different applications. One thing that might or might not be clear at this point, that this approach for generating these models, large science models or digital twins, is actually not tied to any one application. It is actually very general and very broad. And in the interest of time, I'll just talk about one more thing, or maybe 1.5 more things. One more thing, the half thing is this. You can actually take a patient database, like market scan database, which has millions of patients, but it's kind of proprietary. So not everyone can use it, costs like hundreds of thousands of dollars to use per year. But in their licensing terms, which we found out because we tested that theory, they don't say that you cannot share models. So what we did was to submit it to this competition, a giant digital twin learned from their data, and that is public because their license that we signed doesn't say we can share models. But the point is this patient data that was generated from this digital twin is actually indistinguishable as a database from their data. If you try to build a classifier that tries to separate out their data from the phantom patients, you get an AUC about like 0.6 or even lower. But they have almost no correlation. So, but you can make the data public. That accelerates, that has lots of different possibilities, starting from having a public high resolution data to kind of looking at how you can have big personalized digital twins to extend human, like patient health records in the future. There's an unending number of possibilities. So I will end really with one other application, which is under review right now. We call this the algorithm Veritas. I'm not as good as DARPA in coming up with acronyms, so it kind of might seem a little bit contrived. So vetting response integrity from crosstalk and adversarial surveys. Basically, it's an algorithmic lie detector. Can a generative AI tell if you're lying, particularly in structured interviews? The application we looked at was malingering in PTSD. So it turns out that some patients fake PTSD symptoms. It's not always that they are trying to commit a crime. Sometimes the fact that they're faking a symptom is a symptom of a different disease. Notwithstanding the case, we need to know whether someone has PTSD or not because the treatment is different. So what we did was we had this validated database of responses to PTSD questionnaires, and we made a digital twin out of it, a QNet, which can now tell if someone has been asked a random sequence of PTSD questions diagnostic questions, it can predict what your response should be better and better as interview progresses. And if somebody is lying, then their responses, it will consistently deviate from that, and you can run a statistical test on it to determine if they are actually lying or not. So how good is that? That is actually extremely good. It has an AUC of 95%. It has 10 to the power 25 different possibilities that can happen. So more than the number of stars in the observable universe. So you cannot really memorize it. And we looked at 3 different data sets. We looked at a VA data set where people, where patients had PTSD, and it's about 95% where the algorithm said, yes, they have PTSD. Then we went and looked at a survey, a qualitative survey of 300 people, 200 in the US, 100 in the UK, and asked them, they didn't have PTSD or any mental health condition. We asked them to try to beat the system. And then we looked at psychiatrists, forensic psychiatrists, who diagnosed PTSD patients, and asked them to do the same. And here were the results. They were like 210 were actually able to beat it, 6 were able to beat it in the UK, and 1 out of 30 forensic psychiatrists were actually able to beat the system. So you can go and take this test, actually, and try to beat it yourself. Do not do it if you have experienced PTSD or any other mental health condition. But if you have not, try beating it and see whether that works. Your actual credentials will not be recorded, but your responses will be in our database. Okay. And that's pretty much it. So the future vision here in our research group is really use keep developing AI, democratize AI for its power of social good, transform biosurveillance, transform early diagnosis, transform modeling of complex systems. And one last point I want to make is impact of our research and populist discourse on AI. We were, I'll come to that in a moment. There was an interview that Walter Isaacson, who is the biographer of Elon Musk, had an AI interview on which we were kind of featured, given a lot of talks, a lot of news articles written on it. And this was a paper I didn't talk about, which was predicting crime, which is actually using some of these ideas, but in a slightly different way, trying to model stochastic processes and building up equally, trying to get around the problem that, or one of the problems of learning a large-scale neural network is kind of training it with backpropagation, where you have a decay of memory. So if you have a long memory process, then that doesn't quite work. So we kind of developed a framework for addressing that and applied it for predicting crime, and the results were pretty good. It was like 90% accurate. This is what we had.
Did you see that new article about, there was a study about AI having the ability to predict crime?
Yeah, dude.
It was like a very high accuracy rate.
Yeah.
How can we justify just letting people go out there without the aid of this AI when we know that it has a 95% efficacy rate? Do you know that the human soldier, the human police officer, at their very best, does not quite approach 90%?
Yeah, right.
This is going to be better than anything.
Yeah, man.
We'll stop all the violence.
There will be no more crime.
Do you imagine if we allow those motherfuckers? Like, find that story. That story reads like the beginning of that Minority Report movie.
Yeah.
AI algorithm predicts future crimes 1 week in advance with 90% accuracy. What the man? We're in trouble.
I love the picture they picked for it.
Yeah, it's like a dude with a hood on with no face with his hands on the Minority
Report. The Grim Reaper.
Okay, so that's pretty much it. That's my talk. So feel free to ask questions. Yes.
Questions? Yes.
Is that okay?
Yes.
Thank you very much for your presentation. So I have a few questions, but I'm going to ask only one question. Yes. So I'm going to ask a question about like IPF, AUC curves that you have shown in one of the slides. And you discussed that, can you show that slide, please? So you said that for different subsets, you have shown like the ROC curves. And it seems for all of them, you have very good AUC. So I appreciate that you have shown like 3 patients A, B, and C later. But for this, my question is regarding like feature importance analysis. So did you do any feature importance analysis for different subgroups to see if like a specific feature is going to be the most important for all of those subgroups? Did you do that?
So it depends on what you mean by feature.
So I mean,
if you look at this level, where you look at the ICD codes,
yes,
then you can do a SHAP analysis.
yes, that is what I mean.
It's completely useless. Because you're saying that, so we know already what the risk factors are at the population level. That doesn't help us. I mean, it helps to write papers. It doesn't help the clinician at all. Like COPD would be at the top, because for many people, that is important. But if you're looking at an individual, it might not be. So that kind of like a global SHAP analysis, actually it's there in the paper.
Okay.
But it's not very useful.
So I think he's asking for group level. So
yes.
At different groups. When you
because you had for different
for
subgroups.
males and females, for instance, or UCM males or something like that.
What happened to my computer?
The time's out.
You run out of power? I guess I did. So no, we didn't do it at the group level, but it can be done quite easily.
Yeah. So that would be very interesting.
That would be interesting, yeah.
Yeah, because sometimes, I mean, there should be some like, probably, this is my guess, there should be some like confounding factors that can be just revealed throughout that process of doing a feature analysis for all of the subgroups.
Yeah, we did do a very detailed feature analysis. We haven't done it for particular subgroups.
Okay.
One of the understanding was, and you'll see that when you look at the paper, I can send it to you if you have trouble looking at it, is they seem to be not saying anything different than what we already know. Like what's very most important? Well, cardiovascular diseases are most important. Yeah, we already knew that. It doesn't really add anything to our discussion. And although we didn't do it based on subgroups, but that'll be very easy to do it and check whether we get something by doing that. But that maybe we can actually do that.
Yeah, I would be curious because you said that males are more at risk, and I wonder just for males.
We did it for males and females.
Oh, you did it.
Yeah.
Okay, what was that?
There are some small differences, but again, not something that makes too much of a difference in understanding. But there are small differences. If I can bring this up while you guys keep asking questions, I can probably show it.
Yes.
That's there in the paper. The males versus females is definitely there in the paper.
Okay, that sounds good. Any other questions from here? Oz?
Yeah, so for the, I think it was the last one, right? So developing as a PTSD model based on, I think it was based on like several surveys already, questionnaires. So the PTSD, I'm not an expert, but it requires like a previous trauma and then some sort of like tests. These are mostly like a patient-reported information. So basically, the outcome in the model you train, outcome is based on patient-reported results, which varies.
Okay, that's the question.
Yeah, which varies from even time to time. So even like if you do the questionnaire to same patient today and tomorrow, probably it may lead to different results. And when a model learns all those like the patterns of the answering to those questions, and so what I think the final model was like with 0.95 AUC. Was there like a reliability check done this for basically a same test is given to the same patient, whether it says both like one says he's lying, one says he's not lying.
So the question is that there can be different claims. So yes, psychiatric evaluations there's no blood test for that. But I'm not saying you're saying that, but if someone is claiming that no psychiatric diagnosis is worth anything, that's a bigger problem to address. Let's not assume that. Let's assume that if you are sitting with a psychiatrist for a couple of hours, they'll probably be more or less accurate to know if you have PTSD. They're just not going to ask you this question. They're going to ask you, talk to you, they're going to talk about your experience, particularly if they're from VA, they're veterans, then that will be very well validated. So the database on which this is trained is a database like that. It's not like asking people questions for 10 minutes. It's like they went through like about 2.5 hours, 3 hours interview to ascertain that they do have PTSD, right? So that's one. For the other ones, there is some kind of, the work is not complete, it's still ongoing. One of the reasons is that when we ask people on the prolific surveys, try to beat the system. Make sure you don't have PTSD. I do not know that they don't have PTSD. Other than they said they did, right? But they tried their best and they couldn't really beat the test, most of the 95% of the people. And whereas 95% of the people who we know have PTSD, the algorithm did say they have PTSD. So you have let's pass people who have PTSD, we know have PTSD, doesn't let pass people who are most likely don't have PTSD, but there's some still uncertainty there. So we have to actually do a proper controlled trial on this. Again, the other data set we already had, the prolific one cost me like $5,000. You can do that easily. Whereas if you want to run a clinical trial, it's like NIH R1 grant, which now have to go and convince a study group that needs to be done. This needs to be done. There are also ethical questions come up. Should we do it at all? That's the main question that comes up, which really bothers me. And here I'm kind of getting into the rant phase. And that really bothers me because that should not be a question that has been asked. They should be done at all. But they should be rolling it out. That's the ethical question comes next. But because there's an ethical possibility of an ethical question, I will not even interpret the science. That seems, I don't know.
Maybe you will combine your lie detector with the PTSD. If they pass the lie detector, then they can take the PTSD test. That's interesting. Do we have any questions in the chat?
Not in the chat.
Okay. So in that case, I will ask a quick question.
Yes.
So, I mean, this is really exciting. I think it opens up a lot of possibilities to be able to better screen the patients for potential problems. For many diseases, you gave great examples. And I really, really like the possibility of its use for clinical trials. That's exciting. For the cases where before they can make a final diagnosis, they need to do, for instance, some kind of an imaging workup. Are you worried that just, anytime you do an imaging workup, just, they are bound to see some stuff. There could be false positives,
right,
or there could be some abnormalities. Do you think that will cause just, further follow-ups and further problems?
Right. So that's a very important question, right? I had this bright idea of after the paper came out to try to discuss this on Reddit, and people were really nasty and rude. I mean, that's expected. It's internet. But the main point people were making was IPF is a really rare disease. So we shouldn't do this at all because yes, there'll be 1 in 67 number of people who are flagged. 1 in 67 would be a positive case, and you're doing like 60 or 65. You are doing chest X-rays, HSCTs, or whatever, which are false positives. So is that doable, right? So we actually calculated that. Like, what is the risk for cancer due to exposure to HSCTs? And also that's one metric. The other metric is we already do some kind of not universal testing, but something close for lung cancer.
Right.
So is the situation
for
worse?
Smokers, though, right? Yeah.
Yes, true. So maybe we cannot do this universally immediately, but we can do it for smokers. And remember, the data set that we tried this on, the Z-core part, is really bad data, right? It's like EHR data, extremely sparse. Some people might have like 10 data points. I mean, it's very, very sparse and noisy. But we can add it to familial things. Someone has IPF in their family, then they have a 15% chance of getting IPF. You add that, the thing goes up way higher. Basically, you're enriching the set. Right now, it is about 1 in 67. But if you cut down on whom you're going to run this, you can boost it up to 1 in 30, 1 in 20, 1 in 10, where it becomes actually better than breast cancer screening or prostate cancer screening. And the other metric, as I was saying, to look for is NNS, number needed to screen. So how many you need to screen to save one life? Yes. Right? So you can show that even with the 1 in 67, if you can detect it that early, what are the odds of getting a lung transplant? If you carry out that analysis, you will see that it's really kind of the same as lung cancer.
That's good. Let me check. We are out of time, Mustafa.
Okay, that's fine.
Sorry.
I have so many questions.
I know. Just you can follow up with him by email. I want to thank our speaker one more time, and then thank you for all attending.
You can actually ask questions. This is lunch.
We look forward to you attending our next AIIA seminar. Have a nice afternoon.